Good evening, everyone. Thank you. I'm Audrey Bilger, president of Reed College, and I'm pleased to welcome you to our Convocation. Each academic year is a fresh start, and this year we are all mindful of what a privilege it is to be together. I have to say, I got choked up seeing you all here, and that's going to keep going, I think. We are also acutely aware of the responsibility we have for one another. Thank you for wearing face coverings and following safety guidelines. On behalf of Reed's faculty, staff, and continuing students, I am delighted to welcome the class of 2025 and our transfer students, along with family members and friends. I'm also very happy to welcome to campus second-year Reedies who were unable to be with us last year. We are so glad you're here. Orientation Week is all about finding your bearings. There are the literal compass points. Cross Canyon is to the north, PAB to the west. The Great Lawn, where we are right now, is on the south side of campus. Mount Hood overlooks us from the east. Once you get a sense of which direction you're moving in space, you can give some thought to your metaphorical compass points. What are your values? Where is the North Star that guides you when the going gets tough? Last week, I enjoyed a meal with a group of students who share my excitement about meeting you all. Our house advisors and orientation wayfinding leaders. Let's hear it for the HAs and OWLs. I know you've all been benefiting from their leadership and guidance. At dinner, we were talking about what leadership means to us, and we tried to sum it up in one word. The words we discussed were powerful: empathy, compassion, kindness, vulnerability. I know from experience that Reed students take these words seriously. This is a community of care. As metaphorical compass points, empathy, compassion, kindness, and vulnerability can assist you greatly on your college journey. These are all concepts that contribute to a culture of connection. Empathy, compassion, kindness, and vulnerability involve sidestepping the ego and joining forces with those around you. I hope that knowing that you are surrounded by caring Reed students, staff, and faculty will help you feel supported as you embark on this year. You will experience much and will grow a great deal during your time at Reed. In and out of the classroom, you will explore and discover, create, and appreciate. It is important that you know that your presence here matters. We chose you, and you chose us. We belong together, and Reed will be an even better place because you are here. I look forward to getting to know you and engaging with you in the coming weeks, months, and years. Enjoy your first days at Reed, and if you get lost at any point, look around. There's always someone who will help you find your way. Thank you.
What a glorious day in Portland, Oregon. Good evening, I'm Roger Perlmutter. It's my privilege to chair the Board of Trustees of Reed College and to add my welcome to that of President Bilger. We're so glad you're here. Now, you might reflect for a moment on what in the world the Board of Trustees might possibly do. I reflect on that every now and then. But I can tell you at this time last year, the Board of Trustees was deeply concerned about a critical decision that had to be made. Would we, in fact, have on-campus classes? How would we manage them? How could it be done in the face of this extraordinary pandemic? And there were many who argued to us that the idea of having people between the ages of, let's say, 17 and 22 in this kind of environment and trying to ensure testing and social distancing was sort of contrary to the laws of nature, if you know what I mean. The trustees were concerned, but in the end, the trustees decided that the right thing to do was to focus on our educational values and the opportunity to give gifted students a chance to explore important areas of knowledge in depth, working with talented and experienced scholars. That's what we do here. We set up a testing regimen, or rather the staff of the school set up a testing regimen. Tents that you've heard about, earth, wind, and fire, outside that enabled students to study. The faculty came forward, the staff came forward, and most importantly, the students came forward. And I can tell you that throughout the pandemic, the safest place to be from the standpoint of susceptibility to infection was the Reed College campus. And that was because of the efforts that our community made to help one another and to ensure that our values, the things that are important to us as we pursue the cause of gaining a deeper understanding of the world, that our values remained intact. You've joined a very special place. You're all very special people. It's a privilege to welcome you to Reed College. Thank you.
Good day. Hello, everyone. My name is Miljan Trulov. I'm the Vice President and Dean of Admission and Financial Aid here at Reed College, and I'm a real person. I don't live in your computer. So today had a really interesting vibe to it. This was probably the most excited I've seen parents to let go of their seniors who they've been in close contact with for the last 2 years, and so that has its benefits. So first off, I'm going to share all the really important information about the class, and then I want to share a bit about my thoughts with your experience coming here to Reed. You are Reed's largest incoming class. You constitute the largest number of students of color here at the Reed community for an incoming class, and again, you have traveled further to arrive at Reed than any other student has traveled to college. You come from all over the nation and all over the world. In fact, you represent 45 states, 19 countries, and in ascending order, the most common name here for our first-year students is Sophia. So all 10 of Sophias, please stand up and Sophia, nice to meet you. Right after that, Isabella and William. And it is a 6-way tie for the third most common name: Daniel, Elizabeth, Ella, Julia, Lucas, and Zoe. Let's give them a hand. So some of the most common questions that I've received today, yes, Dr. K and I really do get along. No, neither of us are really that funny in person. And everyone you meet will be taller or shorter than they look in Zoom. So late March 2021, you remember that? You all started to hear rumors that the world might shut down, that your school would shut down, and now it was becoming, in 2020, March 2020. And now it was becoming a reality. But it wasn't that bad because, as you know, many of us would come back in a couple of weeks, maybe a month, 2 months tops. But your teachers frantically got homework assignments, put homework together to keep your mind sharp during this very short time. You discussed using Skype or Google Video to stay in touch because Zoom was not yet a verb, like Google or Kleenex. And you certainly didn't expect this during your senior year. You certainly didn't imagine this would impact you, or impact prom, or Sadie Hawkins, or snow days, homecoming, Spirit Week, any of the ritual events that you would normally have in a given year. And this year was very different. And it's certainly not what you had imagined your senior year would be like for the better part of your 16 or 17 years of life. You imagined something very, very different. Not only all that, but now you also had to choose a college. You had to pick a place where you would spend the next 4 years of your life after your senior year. Did you all know that by the end of one's junior year, students usually have their top 10 schools already in mind? And then by the time that the school year begins, you've usually narrowed that list, but it's a result of having visited anywhere from a dozen to 24 schools. You've usually taken the ACT and SAT, and as a result, your names are sent to a lot of places who are reaching out to you to initiate the college process. And now you had to initiate everything yourself. And because you didn't have an opportunity to tour schools for the most part before this all became a reality, instead you sat by your family computer, or your tablet, or your laptop, or your cell phone, and you watched a lot of videos and a lot of Zoom sessions. That is a very interesting way to figure out where you will be for 4 years. So one of the things we endeavored to do was to help you understand the Reed experience through whatever means we had. And we were so glad and happy to meet you online, to talk with you in person, and when campus opened up, about half of you were able to experience our college community. That means about half of you are attending Reed sight unseen. So what do I think you learned from us during that time? I think you learned how to find your college home online, but I'm hopeful that you learned how we will feed your academic curiosity. I'm hopeful that you're able to see how deeply your classmates think about you and care about this experience as much as your faculty and as much as the staff. I'm hopeful you learned how the Reed community manages to live out its values despite the pandemic, and thought to yourself, I'd like to be a part of that. More than half of you are coming to Reed as the first and are the only person from your high school this year to be here on campus. Think about that. When I went to college and it was a small place, there were 2 other people from my high school who were there. More than half of our students here are the only person from their high school, and they made an intentional decision to come here and not anywhere else, and many of them from across the country. They are trailblazers. You are all trailblazers. To our current Reed community, today we are welcoming caregivers, community advocates, artists, musicians, poets, aerial circus performers, falconers, camp counselors, filmmakers, YouTube channel owners, business owners, essential workers, and many other identities. Our incoming students are enthusiastic about improving their communities. They're involved in coding, manga, rockets, ultimate frisbee, crochet ghosts, railroads, sourdough bread, paper cranes, zines, beekeeping, Greek mythology, robotics, and countless other subjects. And so, as veteran community members, I think it's incumbent of us to welcome our newest community with open arms. We are here to tell our incoming class and our new community members that they are not alone, that we have been waiting for them, and that we're glad they're here, right? For our new community members, for each other, I want you to realize, for each other, that community is in large part what you create. I invite you to make welcoming spaces. I'll tell you 5 things. Make welcoming spaces for your classmates. I invite you to share ideas and make spaces for others to share ideas, for folks to embrace difference. Make space for your classmates not to be perfect and not say the perfect thing. Because if you feel welcomed, I think you will give thanks. If your ideas are heard, I think you'll listen to the alternative. If your difference is embraced, you are welcome to share. If you are not perfect, I think you'll have space for self-reflection. And if you don't say the perfect thing, I think you will be gracious when you give feedback. All that was accomplished at Reed last year with our current community, this didn't happen by accident. This didn't fall to us living out our mission during a pandemic. Every person here had to work hard for it. Last year, we worked hard for it. And we learned so much. We learned about communication. We learned about negotiation. We also learned how to unmute after you start talking on Zoom and I never got the hang of that. And this year, we're gonna do it again. This year, we're gonna do it again with your participation and your enthusiasm, because as individuals, you came to Reed, but now you are Reed. Welcome to Reed, Reedies.
This thing isn't nice to short people. Hello, my name is Alondra Loza, but you can call me Aloe. I use she, her pronouns, and I'm your student body president for this year. It is both my honor and obligation to welcome you and your loved ones to your first year at Reed, so welcome. This will be the first year we've all been back in person since fall of 2019, and that's a terrifying thing to say. Being a freshman at Reed has been a difficult undertaking for as long as this college has existed, even without a pandemic looming over us. I remember going into my first year, I was struggling with many of the same things that are probably racing through your heads right now. Will I get along with my roommate? What if I don't have anything nice to say in class? How am I going to balance working part-time while also maintaining good grades? All these challenges are givens in higher education. There are always things we expect going into a place like this, but for others of us, there are additional obstacles that can make existing here feel close to impossible. Reed at times can be an uphill battle. The same factors that make others' experiences here seem seamless and insular can be what make you feel out of your element or develop imposter syndrome. This is for an amalgamation of reasons: where you come from, whether or not you're a first-generation college student like me, and most notably, how you identify on the basis of race and class. This college, like all institutions, works within a state that is created and maintained through white supremacy. At times, the way things are run here can be daunting, and there will be moments during your time where glimpses of Reed can make it obvious that a certain type of student was framed to be its default population. However, I'm here to remind you that you all deserve to be here. Each and every one of you has the right to sit in the very uncomfortable chairs probably that you're sitting in right now. Being chosen to attend this school means that you are in the place of someone else, but that's because you've earned it. Although at times this college's promises of pedagogy and moving towards more reflective and changing discriminatory policies can seem like they aren't working, I want to tell you that Reed is far from perfect. But that's where you come in. College comes with a series of freedoms that some of you may be used to having, but most of you are not. These freedoms means you have the opportunity to make your choices worth something. You all represent the next generation of Reedies, and that means you are bringing a new era of ideas and the power to shape campus culture into what you want it to be. And this all holds true, especially now during a global pandemic. Yes, it's scary to think you'll be entering a school that has existed with its long-standing traditions for over a century, but as a junior who was here just short of a year before COVID changed everything, I realized that even when taking into account the changes of restrictions and social norms, what remains unwavered is Reedy's commitment to be together. Starting next week, we will be entering a time where there will be 3 generations of Reedies who either have only known this place through their computer screens or as something that for me feels like a fever dream away. What has survived is our commitment to make the choice of investing in each other semester after semester. I especially understood this during one of the toughest moments of the last academic year. Some of you may be familiar with the snowstorm that affected Portland last year. Needless to say, the city was not prepared. This meant no Wi-Fi, no power, and for a lot of us, no heat for up to 10 days. During that time, my housemate and I had to walk a mile in deep snow along unplowed roads to campus to use the internet to find a warm and safe place with water and internet. It was then that a senior Reedy offered my roommates and I help before even giving us their name. The community had put together an entire spreadsheet where community members, including faculty, staff, students, and alumni, were offering other Reedies places to stay until the heat came back on. Even after all the challenging experiences that have made up my time here at Reed, this moment reminded me of what truly makes this school, the collective community, which includes the faculty, the staff, but most importantly us, the students. You'll soon come to realize how close we are as a student body, and you will likely feel familiar, you will see familiar faces around you. Most of them are in this tent today. That's just what happens when you go to a school that's as small as this one. And that way, we are all responsible for each other in the fullest sense of the word. So I implore you to contribute to the strength of your community, to listen and to learn from one another, to take criticism and to expand your perspectives, and to be aware of your privileges, and to uphold Reed's values of togetherness, compassion, and resilience. After all, it is our mutual investment in each other that transforms us from students of Reed into Reedies. So I welcome you to your new school, to your new home, but most importantly, to your new community. Thank you.
Welcome. I'm Kathy Olson, the Dean of the Faculty and Professor of Psychology. I have the great pleasure today to introduce our speaker, Sarah Schock, Associate Professor of Biology. Sarah has taught in biology here at Reed for 10 years, joining Reed's faculty in 2011. She was an honorary lecturer in Uganda after completing a postdoctoral training at Lewis and Clark College and the University of Texas at Arlington. Sarah shared with me that one of her parents was an immigrant and that she was the first in her family to graduate from college. She got her BA in biology from Earlham College before earning her master's in zoology at the University of Florida and her PhD in biology at Indiana University. In short, she has an extremely impressive research program at Reed, one that has national recognition, but also importantly, it's important because of the impact that she has on her students here. She estimates that she's mentored about 70 or more undergraduate research experiences. Many of these have resulted in student co-authored publications, around 15 papers with over 20 student co-authors. Many of these are on topics that aren't even our main areas of interest, but they're ones that students were interested in, so she was involved in that research with them. Sarah's mentoring of students and her involving of students in high-level research is not restricted to publications or presentations, but rather these publications are the culmination of a long series of research experiences that she has with her students. These begin in their first years in college, when they're getting involved in serious research at that point. And then they go into higher level classes. They do summer research experiences with her and senior thesis projects. This research is funded by the college, and it's often funded by significant grant money that Sarah has. She's had Fulbright, Murdoch Teaching and Research Awards, and a prestigious NSF Career Award. Let me just give you a little flavor. She'll tell you more about lots of things in her talk, but give you a little flavor of some of her work. Sarah's worked as a biologist since the summer of her junior year of college. She's worked in habitats all over the world, from uninhabited desert islands in the Sea of Cortez to papaya swamps in Uganda to frozen lakes in New Brunswick. Her work has considered large organisms like giant venomous rattlesnakes and small ones like microscopic zooplankton. Her most recent paper was the second in a series from a multinational genomics consortium that Sarah formed in East Africa. The consortium involves dozens of scientists who Sarah has trained in bioinformatics and genomics from more than 5 countries. The 2 papers focus on genomics of a devastating crop pest in sub-Saharan Africa, which is a key partner in the horizontal transfer of DNA between species, one of Sarah's specialties. In addition to being a powerhouse researcher, she takes on important service at the college and more broadly in her organization, in her field, and even in the Portland community. She's involved in major service roles as an elected council member, and her key focus is on increasing equity. In addition to giving research talks all over the country and around the world, she also does a lot of public work where she speaks at, for instance, OMSI, the Oregon Museum of Science and Industry, to tell them about genomics and evolution. She also serves the Reed Department of Biology and is very involved in the campus community, even on the faculty personnel committee. Her energy, her enthusiasm, her commitment to research, her dedication to our undergraduates here, her dedication to read, make her just the right person to provide some remarks at the Convocation today. Sarah Schock, talking about the nature of change.
Thank you so much for the opportunity to speak today and for the kind introduction, Kathy. It is an honor to address you as all of us gather here to celebrate the beginning of a new academic year. Convocations actually aren't really celebrations as much as they are a call to come together, a theatrical call to arms in this case, but the arms are pens, paintbrushes, and microscopes. Specifically, since we are heeding this call in the midst of an ongoing global viral pandemic, I want to talk to you today about the nature of change. I will use the first part of my talk to talk about change in terms of biology, and in the second half, I will talk about the learning project we are embarking on together as a form of change. Then I will share how, for me, at least, these two are connected. The word project will be important today. It comes from the Latin pro jacere. It's the root of to throw forth. As a geneticist, I study mutation, the essential starting point for evolutionary change. The word mutation has recently become a household term relevant in our lives in ways few might have imagined previously. The word evolution is also a part of everyone's vocabulary, but its technical definition is little known. It is not fraught. Evolution refers to when the frequency of genetic variance in a population over time changes. Fundamentally, learning is also about change. In the time that we have together today, I'd like to do some important myth-busting about mutations, but I would also like to draw your ear to some parallels between mutation biology and the learning process that might help equip us for the year ahead, or help us dust off the equipment we have, or even more aspirationally, help us in our approach to the ongoing project of lifelong learning. Mutations have a bad reputation. There are lots of copies of DNA in our bodies, a full copy in each cell, but the copies are not exactly identical. This is because of mutations, which in the public discourse, in most people's individual perception, and pretty much everywhere other than the Marvel universe, is typically thought of as bad. In reality, the term mutation gives no indication about the effects of the physical change that has occurred. As you may or may not remember from your last genetics lecture, DNA molecules are composed of 4 different bases, A's, C's, G's, and T's. And mutations, changes in the DNA, can be big or small. A 1-letter replacement of an A for a G or a C for a T is the simplest mutation to imagine, and that's something that we geneticists call a base substitution. But mutations can also be big. For instance, an insertion or a deletion of tens of thousands of bases, and in some cases, insertions of DNA that encode their own replication and insertion elsewhere in the genome in the future. This kind of mutation is caused by pieces of DNA called transposable elements. Transposable elements and their cousins, endogenous viruses, make up a huge portion of genomes like ours. So this scenario that I'm describing, where a piece of DNA can jump from one spot in the genome to another, is not only a big mutation, it is a common one. Small or large, there is nothing inherent to a particular mutation that determines whether it will be good or bad. What really determines the effect of a mutation is not the physical change itself, but the context in which it occurs. Yes, context matters, even in a discipline thought to rely on statements of fact and absolute truths. so much so that the exact same mutation can be good or bad depending on when or where it happens, and bad mutations can even become good. Good and bad effects of mutations also depend on perspective, of course. Take, for example, rough-skinned newts. These are amphibians that are covered in toxins, rendering them unappetizing to the common garter snakes that typically prey upon them. But in some cases, there are populations of garter snakes that have evolved resistance to the toxin on the newt's skin, a mutation that is good for the snake and bad for the newt. Another myth surrounding mutations is that when they occur, that they happen for a reason. Mutations do not happen for a reason. Mutations don't strategize. Most mutations are caused by one of two events. The first category of events is mistakes. Mutations are mistakes that are made when DNA replicates in the cell prior to cells dividing. Keep in mind, this is a necessary step for cell division, as each new cell that is produced needs to have DNA in order to go about its business. The second category of events causing mutations are those caused by damage. When DNA is damaged, for example, by chemicals or radiation, these injuries are not always repaired, and damage that does not get repaired before the cell replicates will result in a mutation that will be present in the DNA of the daughter cell. This misconception that mutations must have a purpose or an intention comes up especially frequently when I talk about my work on mutations caused by transposable elements. Transposable elements, the pieces of mobile DNA that I mentioned a few moments ago, are capable of moving around the genome, replicating or removing themselves from one position and reintegrating into another position in the genome in a body. This movement can, in some cases, produce millions of copies of segments of DNA that usually do nothing for the host genome in which they are proliferating. Transposable elements represent a third category of mutation event that is very rarely talked about. People always ask me, but why are they there? This is kind of the same as asking, why are any of us here? Although transposable elements are often called selfish DNA, they don't think, they don't make decisions, and they are not intentional. So where am I going with all this myth-busting about mutation? Despite their bad reputation, mutations form the basis of evolution. Without mutations, there is no genetic variation. Without genetic variation, there is no evolution. Without evolution, there are two possibilities. Cellular life remains as it originally formed, and we are all still single-celled organisms floating around in the primordial ooze. This is a best-case scenario for an evolution-free world. The second possibility is that after cells emerged, when the environment changed, life would go quickly extinct. So, while mutations might not always be good, and in fact, current evidence indicates that they aren't even usually good, ultimately, they are responsible for the vast and beautiful biodiversity that we see on Earth today, from cichlid fish to flying squirrels, bioluminescent diatoms to great green macaws, tall conifers to flowering plants. Mutations underlie specific adaptations, like resistance, immunity, and opposable thumbs. There are coloration adaptations like camouflage to avoid predators, skin pigmentation to provide protection from ultraviolet light, or brightly colored petals to attract pollinators. My favorite human adaptation is the formation of the placenta. Placenta foster nutrient transfer to developing embryos. It's especially interesting because the formation of the placental tissue, which is the first organ that forms during development in humans, depends, in part, on genes from the ancient integration of a virus into our genome. This is called domestication of a virus. This is a mutation. It's a big one. And it happens to have a big effect. In this case, the proteins encoded by the genes originating from the virus are called syncytins, and they facilitate cell fusion to help with nutrient transfer and also act as immunosuppressants. Immunosuppression? That sounds bad. But remember the importance of context. In this case, the transition from egg-laying mammals to producing live young within a mother's body required a mutation that prevented an immune response to the developing embryo. This kind of change, this adaptive mutation, resulting from a virus integrating into the host genome, has not only occurred once during the history of life, but repeatedly, independently, in many different lineages at different times. This parallel evolution, in the placenta even, is one where there are dozens of examples of the integration happening over and over again. This has happened with other viral genes for other traits that we consider the hallmarks of our humanity, like brain development. In addition, perhaps ironically, the source for many of the genes that underline our ability to mount a quick and nimble immune response or to stabilize our genome when it faces mutations are from viruses. So while we may feel hemmed in by the various precautions and sacrifices and delays that we have experienced during the SARS-CoV-2 virus, it's undeniable that viruses and humans share an intricate, entwined evolutionary history. Context matters. In fact, parallels between the study of evolution and coevolution in particular are echoed in many other disciplines. The poetic metamorphosis of a Greek sailor as he travels among islands longing for home. The way new patent descriptions refer to previous inventions, thereby reflecting a phylogeny of innovation. The introgression of one language into another when two cultures come into contact. These are examples of patterns we see in evolution and patterns we study in many disciplines here at Reed. What are the connections between mutations, evolution, and the learning project we are starting today? Like the evolutionary change we've been talking about, learning is dynamic and it depends on context. If one were to learn everything there is to know about a given topic and then stop, content in that accumulated knowledge, lulled perhaps into a self-satisfied sense of completion, one would be at best missing out and at worst ill-equipped for the future. For knowledge also depends on context, and new observations, new discoveries, and new circumstances change what we know to what we knew every single day. This requires that we update, read, discuss, unlearn, practice, and explore almost continuously. As a professor, I feel this acutely as I dare not teach what I knew about genetics and evolution last year to all of you this year. What we know has changed, context has changed, and we have changed. Another lesson from mutation biology for learning is that size does not correspond with impact. Small changes can have big effects. Small moments in the studio or the classroom or the laboratory can open up a new hypothesis, pivot a debate, reverse causality, or change your mind. While it is true that it helps to read and reread the carefully curated articles and chapters and books on a given topic, it can be a comment from a friend outside of class, a question scribbled in the margin of a used textbook, or a failed experiment that really opens up a new idea. A third lesson from mutation biology for learning is that while the past may set the stage for the current moment, it does not decide the future. As learners, we all bring our backgrounds and our past experience to this moment, but how we interact and where we go from here is not predetermined. Many of us may even wonder how we got here or if we belong here. I wonder this myself all the time. But I remind myself that this learning project will thrive with me in it regardless of whether or not I feel like I belong. And in fact, the perspective I bring when I am least sure-footed in my knowledge can be a big contribution, that it can generate the most open, creative, expansive moment where I can catapult or be catapulted into new knowledge. The term we like to use is discovery. Discovery is advanced learning. Advanced learning isn't about remembering all the facts or the dates or the formulas. Advanced learning can take several forms: the making or discovering something that no one has ever known before, or changing your mind about something as new discoveries are made. Who do I think about when I think about changing people's minds or when I need to be reminded that I belong, even if I start to feel my otherness? I think about Barbara McClintock, the grandmother of the study of transposable elements, which you now know are basically viruses, but they're already in the genome. Barbara McClintock was a geneticist born at the turn of the last century. She took her first college course in genetics exactly 100 years ago this fall at age 19, even though at the time she wasn't allowed to major in genetics. That class was the beginning of her learning project and a career as one of the most influential and revered geneticists in history. McClintock studied mainly corn genetics, and her work on maize actually yielded a number of landmark discoveries that might have been big enough to earn her the Nobel Prize. But it was her riskiest discovery. It was the observations and explanations of events that were the biggest departures from her knowledge that later earned her the Nobel Prize. Picture it. It's the late 1940s, several years before the helical structure of DNA has even been deduced from the famous Photo 51. People, of course, know that DNA can be inherited when organisms reproduce. This notion, in some sense, had been understood for centuries by the original geneticists, farmers. But the dogma at the time was that the movement of DNA was always vertical, from parent to offspring. But what McClintock observed was evidence of mobility, and more specifically, mutability of DNA within a genome at certain loci. The word loci is plural for locus, from the Latin for place, spot, or locality, and in this case refers to a spot along the DNA. Importantly, the mutable loci she saw appeared to regulate the expression of genes. That is, when these mutations occurred in the genotype, they changed what the corn looked like in the phenotype, generating colorful speckled patterns in the kernels. Gene regulation was a huge discovery, as it explained a huge, looming question in biology at the time. How does an individual organism develop into a complex structure with many different organs and tissues when more or less the same DNA is in each one of its cells? If genes were like beads along a string, what determined which ones were turned on and which ones were turned off in a given cell to make green feathers on some parts of the wing, red feathers on others, not to mention a liver, a claw, and an egg? Nowadays, the topic of gene regulation is an entire course at Reed. But in the 1950s, it was not known or understood how it happened, or even what a gene actually was. McClintock called these mutable loci controlling elements, specifically to avoid the term gene and that can of worms. Perhaps surprisingly, the question, what is a gene, a hotly debated topic in 1950, is still the subject of debate today. What McClintock was seeing, the mutations, the transposition, it was very much counter to the thinking at the time. And as I mentioned a moment ago, genotypes refer to the genetic material of an individual, whereas phenotype refers to what is visible. The departure from an easily mappable relationship between genotype and phenotype was difficult to understand. At this time, genetic material was thought of as static, a blueprint, if you will, and mutagens were thought of as external to DNA. McClintock's ideas did not fit with what was known, therefore it was difficult for people to accept it when she tried to talk about it. In 1950, she made a bold move. She published a paper with no data, no tables, no photos. It was not because she didn't have enough data, but instead because she had too much. The paper wove together her knowledge, her experience, her perspective, her observations, and helped her put forward a new idea. Not providing a definitive answer to a scientific question, but providing a provocative interpretation and opening a door. Shy on evidence, lacking proof, thin on facts. Her accomplishment was not finishing something, but starting it. As she put it, her idea that, quote, once such loci arise, other mutable loci arise through transposition, which in turn become mutable, unquote, may have been, continuing the quote, carried further than the evidence warrants. Yes, McClintock posed an idea. In science, we usually use the term hypothesis, but she posed it from the very penumbral edge of her observations and knowledge, throwing herself far forward into the unknown. That idea would later be tested and find support in thousands of experiments. It would seed decades of work. It would challenge the simple static one-to-one relationship between genotype and phenotype. It would change the paradigm of how we think about DNA and would help usher in a new era of genetic research on non-genes and the mobilome. At the beginning of this talk, I referred to learning as a project. The truth is, I refer to almost everything as a project, which can be unnerving to my family members who are here, who sometimes just want to call it making dinner. But I think coming to college is a particularly project-like endeavor, most obviously perhaps because it is collaborative and it takes a long time, but also because it provides many opportunities to throw forth. When I was in college, I had trouble deciding between 3 majors, but ended up choosing biology because I thought it could optimize the chance I would work outside. A few years later, I found myself in the middle of graduate school, ostensibly to become an ecologist. But instead of just taking ecology classes, I signed up for all the genetics classes that were offered. In 2001, I went so far as to start an evolutionary genetics journal club for reading and discussing scientific papers for fun outside of class. The only rule was that you had to select a paper outside your area of study, not one on the topic you were an expert on. The club had about 10 members. Only 3 of us were graduate students, and the rest were faculty. No one had to go. No one had to do the reading. But we wanted to learn. One week, a very famous, very established botanist, National Academy member, blah, 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 challenged himself and brought a paper for the group to read that presented some of the first large-scale sequencing data for a species of tree. What a gift. This accomplished biologist was willing to be humble, to hang out at the edge of his knowledge when he could have decided to comfortably stay in its center. He didn't ladle out knowledge. He had joined my learning project. It turned out this tree genome was full of these things called transposable elements, something that at the time was never mentioned in textbooks. I said, "What are these?" And I have not really stopped asking that question since. To conclude, what can we learn from mutation biology to optimize our learning project? We can remember the importance of context and perspective. We can remember that small changes can have big effects. We can learn that few things are simply good or bad, even viruses. We can find good collaborators, teammates, mentors, spaces, and support systems to help us. We can be open to learning in different places and in different ways. We can be open to learning outside of class. We can strive to learn things that have been previously unknown, or we can bump into these discoveries accidentally. We can change our mind, and we can change other people's minds, and we can shift paradigms. And perhaps most importantly, we can take comfort in the notion that in those moments when we feel most unsure of our knowledge, we may be actually the most open to discovering new things. At times, we may be feeling like we're in danger of falling, but that could be our opportunity to fall forward, to throw forth, to make a discovery, to pose a new idea. Like evolution, there is no real end to the learning project. And on the 100th anniversary of the start of McClintock's learning project in genetics, the 20th anniversary of my learning project on genetics, I'm grateful not to be done with it. And on behalf of everyone here at Reed, I look forward to joining you in your learning project.
Dr. Sarah Schock, that was wonderful. And we talk about teacher scholars at Reed. You all are in for treats. Thank you so much for being here this evening. Enjoy this beautiful evening, and welcome to the Reed community. Good night.